Many of you had questions about various aspects of the ecology of the salt marsh. So I just wanted to provide you with some information about the ecology and biology of the salt marsh and organisms that live there. Salt marsh ecosystems are found in temperate habitats, both north temperate and south temperate habitats. Where we find them is along shorelines with soft sediments, sand and muddy type habitats, and they're very, very common, particularly where we have estuaries. And estuaries are those places where fresh water runs into the marine shore. So it's that interface between freshwater systems and marine systems. And then they're also found along open marine shores. As you go to subtropical and tropical areas, the salt marsh habitat is then replaced by mangroves, which are trees which can also live in marine habitats and serve a similar ecological role. Salt marshes can be found around the world, but in each of those places as we go from the west coast of North America to the east coast of North America to South America and Europe, what we find is that different species are found in each of those habitats, but they, again, all serve a similar ecological role and have similar sorts of physiological environments that they have to contend with. When you go to the salt marsh, you'll notice that there's a very sharp transition from the coastal forest and other upland vegetation to salt marsh species. Many people want to know why such an abrupt transition. Well, it turns out that both salt and water inundation are very physiologically challenging for plant species and require very special adaptations. We see water inundation being a real problem for houseplants. If we water them too much, they die. It turns out that salt, which is part of the marine world, reduces availability of fresh water, and those plants need fresh water. Inundation by water reduces the amount of oxygen that's available, and plants also need oxygen. So that combination of having salt in the water and too much water results in only a few plants being able to live in this habitat. Here on the east coast of North America, Spartina alterniflora, or smooth cordgrass, is the major species that we find dominating the lower marsh. It's found in North America from Newfoundland all the way down to mid-Florida and along the Gulf Coast. Along its range in eastern North America, the species composition of this marsh is amazingly similar. So we see very, very similar species all the way from Canada down to Florida and the Gulf Coast. So when we go to a salt marsh, there's this huge amount of biomass in the grass. One of the questions that students are frequently confronted with and have a hard time thinking about is, where does most of that matter in this grass come from? One possibility is light from the sun. B, minerals and nutrients from the sediment. Or C, molecules in the air. Or D, none of the above. What's the correct answer? Well, it turns out it's molecules in the air. And this is something that does challenge a lot of students. Plants are primarily made of carbon. They get that carbon through photosynthesis and the use of carbon dioxide in the air. So again, this is a really hard concept for students to think about. If they were just using all the minerals and nutrients from the sediment, that sediment would all disappear. But we know from our houseplants that dirt in our little pots of our plants doesn't disappear, but our plants grow. So again, this is one of those tough questions that students do have a hard time with, but need to understand if they want to understand that link between all the parts of the ecosystem, how plants grow, and how they use different aspects of their environment. So students are sometimes also confused about grass and food. So let's say that during a class discussion about plant growth, a teacher asks, where do these marsh plants get their food? One student says, grasses make most of their own food, but they also get a little bit of food by soaking up water through their roots. Okay. Which one of the following ideas does the student seem to be missing? 1. A. Water is not food for grasses. B. Grasses do not make their own food. C. Grasses get most of their food through their roots. Or D. None. The student's statement is correct. What do you think? Well, the problem is that water is not food for grasses. Remember, food is that attaining of matter that we then convert into our own matter, or for plants, gaining of matter, which then they convert through photosynthesis to make more biomass. Water is not food for grasses. Let's talk a little bit more about the smooth cordgrass, Spartina alterniflora, and the special adaptations it has, different from other plants, for living in the marsh. Now, one of the things that we talked about that is really important for plants and restricts their ability to live in marshes is that they have to deal with salt. All that salt in there reduces the amount of fresh water that they can basically use in their tissues. Well, Spartina alterniflora can secrete salt through its leaves and has salt glands. If you lick the surface of a salt marsh plant, it'll taste salty. It also has a very thick cuticle, which reduces loss of water, and its stomata, or the little openings for exchange of gas in the leaves, are located in very deep grooves. This also reduces the amount of transpiration, the loss of water that will happen through living plant, so it helps it conserve that fresh water. Now, both of these, this thick cuticle and that secreting salt, make it much less palatable to insects. So insect herbivores are found in a lot of the vegetation above the saltwater in the forest, but you tend to see very, very few insects that live anywhere in the salt marsh. So they're also being protected by these adaptations that allow them to deal with being in saltwater. They also use C4 photosynthesis. Now, C4 photosynthesis is a type of photosynthesis that uses less water for primary production. So it's another special adaptation by these plants. One of the other challenges that salt marsh plants have is that they are covered by water, which greatly reduces the amount of oxygen available. So waterlogged plants, we see very, very low oxygen. Now, if a plant is continuously flooded, it can survive by respiring anaerobically, so without too much oxygen, but growth is very decreased. So what do we see in Spartina alterniflora? It has very special tissues, parenchyma, which are airspace tissues, which move oxygen to the roots for aerobic respiration. As I said, now these plants do have special adaptations and special metabolic adaptations to flooding that help them live in this harsh environment. But one of those most important ones is this ability to move oxygen down into their roots so that they can have regular old metabolism like plants that aren't under oxygen stress. So another question that frequently confuses students. Let's talk a little bit about respiration. So, which of the following organisms release carbon dioxide into the air when they metabolize food? So, any kind of food, sugars, glucose, anything. So which kinds of organisms respire or release carbon dioxide into the air when they metabolize food? Animals? Plants? Both? Neither? Question mark? Well, the answer is both. Students understand that plants photosynthesize, but they forget about the part that those plants have to do the same metabolic work that animals and everyone else has to do. They have to replace their cells, repair their cells, do all the same kinds of living that animals have to do. So they also respire. Let's go back to the salt marsh for a bit. Okay, so when we were out there in the salt marsh, we saw that there were tall plants and short plants of the same species, Spartina alterniflora. And this is one of the things that we wanted to understand. What might cause some of those plants to be tall and some to be short? The taller ones, do they have more biomass? Do the short ones have less biomass? What's the difference, and why might we see this difference? So let's talk a little bit more about what plants need to grow. Now, they need nutrients like nitrogen and phosphorus to grow and build more tissues. The nutrients that they use are absorbed through their roots in the sediment, not from the water. And a lot of people wanted to know what's the difference between sediment and soil. Now, sediment is deposited by hydrological processes. Soil, on the other hand, is generated by geological processes, like erosion. So the sediment that salt marsh grass is in has been moved there from elsewhere by rivers and streams or waves in the seawater. So the sediment that they live in was made elsewhere and deposited there. So they have roots that go into the sediment, and it's from that sediment where they get their nitrogen and phosphorus and other nutrients they need for growth. They also have rhizomes that are underground that extend through the sediment, and then individual grass stems grow up from those rhizomes. So this is much like grass that you see in a lawn, where there's little rhizomes that extend and the grass can then grow vegetatively. So most of their growth is expansion of asexual propagation, right? It's not from individual seeds, but the plant growing and extending and the rhizomes moving into new areas and grass stems growing up from those. You can see these different parts of the plant here in this slide. So we have the green parts, or the stems that are coming up. Those are all that above ground biomass that we see. And then we can see the finer white roots that extend into the sediment that then absorb the nutrients and help anchor the plant. And then there are these longer rhizomes, and these rhizomes then grow through the sediment and new stems then emerge from them. Here's another question about plants that sometimes causes students to have a bit of confusion. So, which of the following are flowering plants? Plants that produce flowers? Broccoli, potatoes, grass, or all of the above? What do you think? Well, the answer is, of course, all of the above. We tend to eat broccoli before it's flowered, but those of you who have left your broccoli in your refrigerator too long will see that it does produce little flowers if you let it. Potatoes, of course, are the underground storage bodies for a flowering plant. And if you go to a potato field, you don't see any of those potatoes because they're under the ground, but the above ground plants, of course, making lots of flowers. Grasses also make flowers. Their flowers don't look quite as elaborate as everyone else's flowers, but they do produce flowers and seeds. So smooth cordgrass can reproduce both asexually or vegetatively from those rhizomes growing and new stems growing up from that, as well as sexually. They are flowering plants and they do produce seeds. Now that sexual reproduction through seeds really contributes very little to the growth and maintenance of a particular stand of grass, so most of that is asexual reproduction. However, seeds and sexual reproduction become very important when you have colonization of a new site. So if there's some disturbance that removes some of the salt marsh grass, then it can recolonize through seeds, and it can also expand into new habitats. You can have new marshes formed from seeds that emerge elsewhere. But the seeds are quite tiny relative to the size of the overall grass that grows vegetatively. If we look within a marsh, there will be local spatial differences in productivity, and these differences are set primarily by abiotic factors. And again, it's those same abiotic factors that make only salt marshes able to live in the marsh, those stressors like how much oxygen there is in the sediment, what the salinity is, and those toxic sulfide chemicals. There's also spatial variation in things like nitrogen uptake and assimilation. And in general, nitrogen is the limiting nutrient for plants. This is why we add it as fertilizer to increase plant growth. And small variation in those abiotic factors are going to influence local differences in productivity. The zonation of species on the shore is also due to tolerance to those abiotic factors, in particular salt, low oxygen, and increased sulfides. So if we take a step back and we look at the whole marsh, we will see a zonation in all North American salt marshes that are driven in large part to species-specific differences in tolerance to inundation, salt, and low oxygen. So Spartina alterniflora is the dominant species, low on the shore. Higher on the shore above it is Spartina patens, a close relative. It's sometimes called sea hay. It was that nice short grass that we saw above it, and it has lower tolerance of salt, lower tolerance of being inundated, and low oxygen. You'll occasionally see spike grass or juncus there, and where juncus will be found will be in little hillocks, little bits of increased elevation, because again, it does not do well with being inundated too long. Now, high on the shore, above the Spartina, we'll see increasingly Phragmites australis. This is a non-native introduced species. It's from Europe, and it is predominantly a freshwater species, but it has tolerance to very, very low salinity. So where we see it is just at that fringe, at the upper part of the marsh. And increasingly, this invader is spreading, and we see it in more and more of our marshes. The Spartina can also be affected by animals in the marsh. Through their biological activity, they have effects on those abiotic conditions in the marsh. So for example, fiddler crabs, predominantly Eucopugnax and Eucopugulator, and mud crabs, species in the genus Panopeus, dig burrows. These burrows will increase oxygen into the sediment, as oxygenated water can then move down into the sediment, and sometimes the animals themselves, their behavior, but usually it's just the construction of how they build their burrows, drive the oxygenated water deeper into the sediment and move it around. Now, mussels are suspension feeders, which means that they feed on particles suspended in the water, and they are moving particulate organics to the sediment. You'll remember that nitrogen that plants need is taken up by their roots, not out of the water. So if mussels are moving that organic materials to the sediment, that can make it more available to the plants. Also, animals excrete ammonia, and this excretion increases local nitrogen availability. So the waste products of aquatic animals is ammonia, which is very, very easy for plants to take up. It's a very easily reactive form of nitrogen and a good fertilizer. Here are the two species of fiddler crab that dominate in our marshes, Yucca pugnax and Yucca pugulator. You can usually tell Yucca pugulator because it has that purple spot on top of the carapace. These are both males. The males have these very, very large claws. One claw is way large. The other claw is very small. Only the small claw can be used for feeding, and that large claw is used for signaling females, signaling other males that this is my territory, and for male-male combat. Females have two small claws. What does this mean? It means that females can feed at twice the rate that males can. So that's how it works. The males can only eat half as fast as a female can. The other crab that we see that burrows in the salt marshes are various species of mud crab, Panopeus. They have firmer claws. They're better predators at eating a wide range of things. The Yucca, the fiddler crabs, eat particulate organics that are found in the sediments. Now, marsh communities generally have higher biodiversity than sandy shores. Sand shifts and moves around a lot, so it's very hard for species to live there. It's hard for animals to live in that kind of moving environment, and it's also hard for plants. So we see much more diversity both in plant life and in animal life in marsh communities. Now, these communities are also being affected by non-native species. Again, high on the shore we talked about Phragmites australis, the reed grass that is from Europe. It can't tolerate very much salt, so we see it only very in the high shore, but it is increasing in abundance and makes a huge amount of biomass. An animal that we are now starting to see in our salt marshes is the Asian shore crab. Previously it was restricted just to cobble shores and rocky shores, but now we're starting to see it more in our salt marshes. So Hemigrapsis sanguineus is now an increasingly an important predator. It does not burrow like the fiddler crab and the marsh mud crabs, but it does play an important role in being a predator, and it also eats algae. So it's an omnivore and can be very disruptive and particularly attack not only mollusks but also other crabs. Ribbed mussels are very common in salt marshes. In fact, they're the dominant bivalve that we see in these marshes, and they're very often very closely associated with the salt marsh grass. Mussels are suspension feeders. That means that they are feeding on organic particles that are suspended in the water column. They have tons and tons of cilia on their gills, which are quite large, and those cilia drive water movement past their gills. Now, as that water moves past their gills, they can use this both for oxygen exchange, for respiration, and for feeding. So when that water moves past their gills, particles in the water adhere to mucus on their gills, and then those fine particles are moved to the mouth and sorted for feeding. If we take a close look at the inside of a mussel, this is a mussel where just one of the valves has been removed, we can see all the little working parts. We can see that there's a very, very extensive gill. There's two of them that lie on both halves of the mussel. The upper gill has been removed, so we can see the other parts of its body. There are labial palps that are really important for sorting particles, and they then move particles into the mouth. They have a foot that makes these protein byssal threads that it uses to attach to other mussels and to the stems of the marsh grass. Mussels then pump water with their cilia on their gills into their valves that brings in phytoplankton and particulate organic carbon. It's sorted and selected on their gills, and then they decide which parts they're going to ingest. And in fact, they only feed on very high quality particles that are within a certain size range. The particles that they don't want to eat on are then rejected, and those are bound up in mucus by the labial palps and parts of the gill into what are called pseudofeces. Pseudofeces are particles that have not gone through the digestive tract. They look like feces, but they're not. These are just flushed from the valves and deposited on the bottom. Now, post-digestion also produces feces and excreted ammonium from their kidneys, or nephridia. So they are putting organic matter that is nutrient-rich on the sediment, both through pseudofeces, the particles that they don't want to eat, and then the post-digestion result of what they did eat, both feces and excreted ammonia. Here you can see the differences between feces on the top and pseudofeces on the bottom. This is part of an experiment that some researchers were doing where they were giving them phytoplankton, green food that they could see very readily, and then particulate, plant matter, and other sorts of organic matter. And you can see that the algae that they put in produced green feces and green pseudofeces. The other particles produced the brown feces and the brown pseudofeces. Pseudofeces in general are a lot looser in construction than the feces, but they produce a lot more pseudofeces than feces. Hopefully a lot of this information now about the biology of Spartina and the workings of the marsh will help you work on revising your models. Good models will incorporate known information that is relevant to the phenomenon, and hopefully a lot of this information that is in this presentation will help with that. Next, let's look a little bit at the data you collected from the field to see if those data will help you refine the models you're developing. So if we take a glimpse at your data from the field as averages, so these are the averages within each 25 by 25 centimeter quadrat that each of the teams collected. On average, the tall grass was taller than the short grass. Yay! There was an average 104 millimeters height in the tall grass and an average 62 millimeters in the short grass. Great. There was no real difference, though, in the number of stems there were. So there was the same number of stems, essentially, in the tall grass, 23 per quadrat, and 24 in the short grass. There was, on average, a difference in the number of mussels. The tall grass had, on average, 4 mussels per quadrat, while the short grass had 1 mussel per quadrat. So there were some differences. So let's look at the rest of our data. So this table represents the mean of various factors that we were looking at across 5 quadrats, the 5 25 by 25 quadrats that we took. The tall grass is in the top row and the short grass in the second row. Let's look at the above ground biomass. There were 43.71 grams on average in the tall grass, while in the short grass there was only 14.92. Now below the mean is the standard error. The standard error takes into account not only the variance, the standard deviation that we found among quadrats, but it also takes into account the number of quadrats that we had. So we can see that there's a very large difference in the above ground biomass between the tall grass and the short grass. Well, let's look at the below ground biomass. That below ground biomass, however, was much more similar. There was still more below ground biomass in the tall grass quadrats than the short grass quadrats. But that difference is far less than what we see in the tall grass versus the short grass above ground biomass. Now that above ground biomass we know is probably driven by the size of the grass blades that were there, because we found a similar number of stems between the tall grass and the short grass. So what else did we find? Well, we talked about the number of mussels that we found on average, just over 5 in the tall grass versus just over 1 in the short grass. But let's look at the mass of the mussels. So the ecosystem effect, the pumping effect, the filtering effect of mussels is far more related to their total mass than to just their number. We might have a lot of small mussels in one, large mussels in the other, but we can again see that there's a very large difference in mussel mass in the tall grass versus the short grass. So the functioning of those mussels would be far greater in the tall grass patches than in the short grass.